Hello students, I am Mohan Kumari. Welcome back to MK Classes for Biology. In today's video, I am going to explain you about the structure of human gametes, that is the sperm and the ovum. So in the previous video, I have explained you about the process of spermatogenesis and oogenesis. At the end of these processes, we could get the gametes. So we are going to study the structure of those such gametes now. So before going to start with the concept, those who are watching my video, please like, share and subscribe my channel. So here we go with the structure of human sperm. So this structure of human sperm, it is very important concept in the chapter human reproduction. So explain the structure of human sperm can be asked for 5 marks question. Here we go with the structure now. Now you can see the diagrammatic representation of the human sperm where it is differentiated into different regions. You could make out the different regions of the sperm, that is head region, neck region, middle piece and the tail region. Let us study these regions one by one. Here we go with the head region of the sperm. So if we can see the entire region of the entire structure of the sperm, head is the enlarged end of the sperm containing the large haploid nucleus. Of course, gametes are haploid in nature, that is condensed chromatin body it consists of in the head region. So and this head, the nucleus, it is capped by acrosome. You can see the end of this head region, it is capped by acrosome. So what is this acrosome? So the acrosome contains hydrolytic enzyme that help in dissolving the membranes of the egg which is required for fertilization. So the egg will be surrounded by different layers or different membranes. So the sperm, in order to release its haploid nucleus into the cytoplasm of the egg, it has to cross these membranes. So this hydrolytic enzyme which is present in the acrosome of the head that help in dissolving these membranes so that it can release its haploid nucleus into the cytoplasm of the egg. So that is required for fertilization. So the next region of the sperm is the short neck region. You can say this is the neck region. The neck of the sperm, which consists of a pair of centriole. So that is the proximal centriole towards the upper side, that is nearer to the egg nucleus, and just below this distal centriole. So this proximal centriole go along with the sperm nucleus into the cytoplasm of the egg. So after the fertilization, that means once the nucleus of the sperm fuses with the nucleus of the egg, that forms the diploid cell now. Now that is called as the zygote. So this proximal centriole which is necessary for the first cleavage division of this zygote. Yes. And the centriole which is just present below this proximal centriole in the neck region, that is connected to the tail filament. So this is about the neck region of the sperm. Let us move on to the next region, that is the middle piece. You can see this is the entire middle piece. So the middle piece of the sperm contains a number of mitochondria. You can see inside that contains a number of mitochondria that provide energy for the movement of the tail. So the tail part of the egg which show vigorous movement. So in order to show this movement, it requires energy. That energy which will be provided by the mitochondria which is present in the middle piece of the sperm. So that movement is necessary for the sperm. It is essential for fertilization process. So the last region of the sperm is the tail region. You can say this is the tail region. This tail region consists of axial filaments. You can see it consists of axial filament and it is surrounded by the plasma membrane. Of course, it is an animal cell. It is surrounded by the plasma membrane. It helps the sperm to swim in the fluid medium. Yes, tail is very essential to show the motility which is required for fertilization. This is about the structure of human sperm, where it is differentiated into head region, neck, middle piece and the tail region. A human male ejaculates about 200 to 300 million sperms during a coitus in 3 to 4 ml of seminal fluid. That is, per intercourse, the male partner is going to ejaculate 3 to 4 ml of seminal fluid that consists of 200 to 300 million sperms. For normal fertility, at least 60% of sperms must have normal shape and size, and 40% of them show vigorous motility. That means highly motile. The seminal plasma along with the sperms constitute the semen. That is, semen is nothing but seminal plasma plus sperms, now called as the semen. In humans, it takes an average of 74 days to form sperms from the spermatogonia. That means in order to complete this process of spermatogenesis in humans, it takes an average of 74 days. The released sperm to the female reproductive tract is viable for about 48 hours. If the sperm count is less than 20 millions, the person is said to be sterile, and that condition is called as oligospermia. Absence of sperms in the seminal fluid. Seminal fluid secreted by the accessory glands of the male reproductive system. If there is no sperm in the seminal fluid, that condition is called as azoospermia. Yes, here we go with the structure of Graafian follicle. So we had studied different developmental stages of follicles during oogenesis. So we had studied primary follicle, secondary follicle, tertiary follicle, and the matured follicle is the Graafian follicle, which is enclosing the egg, or I can say actually it is secondary oocyte. So let us study about this structure. It is also important for NEET. So you can see the structure of Graafian follicle now. So the wall of the Graafian follicle made up of outer theca externa. You can see this is the outer wall, theca externa, and inner theca interna. Outer theca externa, inner theca interna. So inner to the theca interna, that means inner to the theca interna, are 3 to 4 layers of granulosa cells. So here you can see 3 to 4 layers of granulosa cells which appear like a membrane. Here you can see these are the granulosa cells. These granulosa cells which appear like a membrane. Hence it is called membrana granulosa. It is called membrana granulosa. Now, the membrana granulosa encloses a large central cavity. You can see here is the cavity. This cavity is called as the antrum. And this cavity, it is filled with the fluid called as the liquor folliculi. This is the antrum filled with the fluid called liquor folliculi. At the end of the membrana granulosa is a heap of, you can say this end, it consists of heap of cells projecting into the antrum. And this cells are called, this region now it is called as cumulus oophorus. Cumulus oophorus. Now you can see this is the cumulus oophorus. So just present on this is the cumulus oophorus. Present on the cumulus oophorus is the secondary oocyte. We are calling it as egg. Secondary oocyte or the egg. The matured Graafian follicle ruptures, releasing the secondary oocyte. This process is called as the ovulation. So release of the secondary oocyte from the matured Graafian follicle by a process called ovulation. Now, this is the structure of ovum or the egg. Human ovum is said to be non-cladoic or lethal microscopic structure. So non-cladoic means without any shell. All-acetal means without yolk. And it is of course microscopic structure. The ovum possesses 3 coverings. The inner covering is called as the plasma membrane or the oolemma, you can see there. The middle layer is said to be the zona pellucida, and the outer is the corona radiata. Yes, ovum possesses 3 coverings: inner plasma membrane called oolemma, middle zona pellucida, and outer corona radiata. Oolemma encloses the cytoplasm of the egg, which contains all the cell organelles and a prominent nucleus. You can say the nucleus is highly prominent. So this nucleus is called as the germinal vesicle. There is a narrow space between oolemma and zona pellucida. You can see here there is a space between the oolemma and zona pellucida. The space which is called as the perivitelline space. The space is called as the perivitelline space. Fertilizin is a compatibility receptor protein found in zona pellucida. So egg which will be provided with 3 membrane. So the inner one is oolemma, next to oolemma is zona pellucida, outer to zona pellucida is corona radiata. This middle zona pellucida, which consists of a receptor protein which is called as the fertilizin. So it is a compatibility receptor protein seen in the zona pellucida. So because of this receptor that facilitate the binding of sperm head to this region. So the uppermost pole of the egg, that it is the site of maturation division, is called the animal pole. So uppermost pole of an egg, that is the site of maturation division. So when sperm penetrate into the egg, it complete its meiotic cell division. That region of the egg, now it is called as the animal pole. The lower pole of the egg, that is opposite to the animal pole, is called as the vegetal pole. So these are the extra information for you, and it is required for NEET. So let us study the differences between these gametes, that is sperm and the ovum. Sperm is the male gamete produced in the testes, and ovum, it is the female gamete produced in the ovary. Sperm, it is a long flagellated motile cell differentiated into head, middle piece, of course neck, middle piece and tail. Ova, which are rounded spheres and non-motile. The sperms have negligible amount of cytoplasm and the nucleus is exceedingly compact. Here, the egg, they have bulky cytoplasm and a centrally located nucleus. Sperms are produced in large numbers. Ova are produced singly in humans. Sperms are not covered by additional membrane. Eggs are covered by additional egg membranes. So these are the important differences between the sperm and the ovum. So thank you. This is about today's video. So in this video, we had studied about the structure of male and female gametes of human. Just keep watching my channel. Once again, thank you.